Hello everyone. For this lab, we're going to talk about ubiquity of microbes. There are a few vocabulary terms that you need to be familiar with because you're going to use them a whole lot in this class. The first one is microorganism or microbe. So we can use microorganism or microbe interchangeably. Microorganisms, those are living things that are too small for your naked eye to see them. And so you would need a microscope to be able to see these tiny microbes. We're going to talk about bacteria, fungi, protozoa, and viruses. And viruses, scientists do not consider them living because they cannot replicate on their own. They must have a host, and so therefore we do not consider them living microorganisms. But they are still under the group of microorganisms or microbes because they're way too small. There is also the term ubiquitous. So ubiquitous means that these microbes, they are found everywhere. An example of a ubiquitous microbe is bacteria. So bacteria, they're basically found everywhere, in the air, on surfaces. They are found inside of you, in your guts, within like on your teeth, even on your hair. There is bacteria on your skin. They're basically found everywhere, even underwater. They're found, underwater in lakes, ponds, oceans. That's what ubiquitous mean. Contamination is another term that you need to be familiar with, and contamination means unintentional exposure to microbes. Meaning, let's say that in the lab you want to go ahead and you want to grow a specific type of bacteria. But instead of the word grow, we actually use the term culture. So I want to culture a specific type of bacteria. Let's say that I want to culture E. coli. Okay. Now, for me to culture E. coli in the lab, and only E. coli, I must make sure that all the utensils that I'm going to use are sterile. Because if they are not sterile, remember how I said bacteria are found everywhere? So if your utensils are not sterile, guess what? They're going to have bacteria on them. And so when you use these utensils, you're going to unintentionally contaminate your sample, and guess what? You're not just going to be growing E. coli, you're going to be growing E. coli and whatever bacteria that was on that unsterilized utensil. Okay, and so this is what contamination means. And so therefore, you are going to learn steps to lower the chances of contaminating your samples in the microbiology lab. There's another term that you need to know, and that is inoculation. Now, inoculation is the intentional exposure to microbes. I want to go ahead and intentionally grow E. coli on a plate. And I'm gonna show you a picture of some of the plates that we use in a microbiology lab. But I want to go ahead and intentionally grow the E. coli on that plate. And so that is known as inoculation. You might have heard the term inoculation. When we use it in our everyday lives, we are usually referring to vaccination, right? We say we're going to get inoculated, meaning we're going to get vaccinated. And so why do they use the term inoculation and vaccination interchangeably? Because think about it, when you get vaccinated, aren't you intentionally exposing yourself to a specific microbe? You are. So when you get vaccinated, they intentionally inject you with a specific virus, for example, so that you can go ahead and start building immunity against it. So again, that's why we call it, we call vaccination, it's basically you getting inoculated. Now, what's another term that I want you to know? Fomite. So fomite is an object or a surface that can basically harbor and transfer microbes, such as your computer screen. Right now, on your computer screen or on your computer keyboard, there are bacteria that are, right there on your computer screen. And your computer screen, for example, would be considered a fomite. Now, only computer screens? Absolutely not. Okay, pens, pencils, your desktop, your desk. I mean, all of these are surfaces that can have bacteria basically on them. And they can be transferred. So let's say you touch your computer, you wipe your computer, did the bacteria, is the bacteria going to transfer from your keyboard onto your skin? Yes, it is. And so that's why it's extremely important that when you work in the microbiology lab, you wash your hands before you start doing any work, because there's going to be a lot of bacteria on your skin. And so to prevent contamination, we ask you to wash your hands before you start doing any work. The term culture is basically growth of the microbes. So when I want to grow a specific type of bacteria, or when I just, it doesn't have to be specific, like if I just want to grow bacteria in the lab, we use the term culture. This is a picture of what you're going to see when you go ahead and try this week's virtual lab. And this virtual lab is going to actually go ahead and ask you to sample different objects to see whether these objects have bacteria on them, since we said bacteria are ubiquitous, meaning everywhere. We want to go ahead and sample different things in the lab and go ahead and see whether they have bacteria on them or not. So let me kind of show you some things that you're also going to get exposed to when you are doing that virtual lab. And the first thing that I want to talk about, those stacked plates right here, we refer to them as Petri plates. So basically, those Petri plates, they're basically shallow plastic dishes, and we go ahead and we pour in each dish, we grow the growth medium. That medium, or plural, the media, it's basically going to act as a surface that has a bunch of nutrients that's going to allow the bacteria to grow on that medium. And so those Petri plates are basically used to provide nutrients for your microbes, so that your microbes can go ahead and grow on those Petri plates. So again, this is how a Petri plate would look like. As I said, they're kind of like shallow. They have a lid right here, so that's their lid, and this is the bottom of the plate. And right here, you guys can see that it's shallow. So what we usually do is we pour the medium inside of that dish, and this is the medium right here. Do you guys see that jello-like structure? This is the growth medium. Now, this medium, it's usually going to have a bunch of nutrition, basically, like a bunch of nutrients to help the bacteria grow. Because for bacteria to grow, for us to culture bacteria, they need food. And so that medium right here is going to provide the nutrients for your bacteria to go ahead and grow. Now, when you look at this medium, kind of looks like it's solidified, and that's because we add something called agar. So agar, it's extracted from seaweed. We add it to the nutrients, and the whole point of adding agar is to solidify the medium. Now, can bacteria consume agar? They can't. And so that's perfect because then you can have a surface for the bacteria to grow on it, and the bacteria cannot basically consume the agar, so you can be able to visualize the bacteria growing on the medium. These plates are sterilized, so when we go ahead and make the medium right here that we're going to pour into these dishes, the medium itself has to be sterile, and this is with the lid on. So we have to keep the lid on until you're ready to inoculate it. So if you're not ready to inoculate it, you must keep the lid on. Why do you have to keep the lid on if you're not ready for inoculation? Because once you take the lid off, it's going to be exposed to air. And as I said, air contains bacteria. Remember, bacteria are ubiquitous. They're found everywhere. So the second you go ahead and you take off that lid, the bacteria in the air can actually go ahead and start falling down onto your medium. And so therefore, you have to kind of be quick when you want to inoculate that surface with bacteria. You want to be quick in terms of the inoculation process. And another thing, you don't want to completely open up the lid like so. So this is kind of, that's a no-no. We do not do that. We usually open it like a clam shell, okay, to prevent, again, any bacteria from the air from falling down on your plate, unless you want to expose that plate to air. So let's say I want to go ahead and expose that plate to air so I can go ahead and culture the bacteria in the air, then by all means, take off this lid and actually keep your plate exposed to air for a few hours, okay? And then go ahead and put the lid back on. But if you don't want to sample the air, then you don't want your plate to be contaminated. So make sure that you open it like a clamshell and close it back up pretty quickly. And one more time, this is how your Petri plate is going to look like. Has the lids that I showed you, has the bottom part, and you always have to label your plates. Why do we have to label our plates? Because you want to remember what did you inoculate on that plate. Okay, so you have to, we always label it with what sample did you use, or basically like what did you sample. So is this a sample of a certain type of bacteria that you inoculated? So is this a sample of E. coli? You gotta write down, okay, this is E. coli. Or were you just sampling a surface? Okay, so did I sample my keyboard? Did I sample my chair? You have to write down what did you go ahead and, you took a sample out of what? You have to write that down. We also put on the date, okay, and you also put on your initials. You have to write pretty small, okay, so you have to write pretty small so that it doesn't take too much space, because at the end of the day, you're going to take a look at this plate, and so you don't want your huge writing to get in the way of your observation, okay? Why do we always label the bottom part of the plate and not the lid? Because the lid can fall off, okay? And if the lid falls off, and let's say you have multiple plates and several of the lids came off, then you're not gonna know, which lid belongs to which plate, okay? But if you label, the bottom of the plate, you are safe. And when you come to incubate those plates, and I'm going to show you the incubator in just a few. But when you come and incubate those plates, you have to put the jelly side, or basically the agar side, towards the top. So the jelly side or the agar side always have to be placed side up. Why? Because you guys are going to notice that the plate lid, okay, the lid usually has it can actually have some water condensation. And so the thing is, if your lid is on top of that jelly, what's going to happen is all of that water, okay, like condensation that you have on your lid, it's going to start trickling down onto your jelly. And water coming down on the agar can actually cause contamination. And so to prevent any of that water condensation from getting onto your agar, we have to place the agar side up. In the incubator, as I said, it's basically right here. This is an image of an incubator. It's basically like a chamber, okay? And this chamber, we can go ahead and actually play around with the temperature settings. So you can go ahead and set it at different temperatures. Bacteria, they usually grow at room temperature. But room temperature, by the way, it's around 23 degrees Celsius. Okay, that's room temperature. Bacteria, they can grow at room temperature, and they will grow, but they prefer a bit higher temperature. So they prefer 37 degrees Celsius. Okay, that's like their preferred temperature. And by the way, 37 degrees Celsius is your body temperature. Okay. So as I said, room temperature is around 23, body temperature is around 37, and actually bacteria prefers body temperature. So we can go ahead and set right here. You can set your incubator to 37 degrees Celsius, and you can see it's the chamber, right, with a bunch of shelves. So you go ahead and you place your agar plates. You can call them agar plates, by the way, or Petri plates, right here on the shelves, and just make sure, right, that you place them agar side up, because you're putting them in a pretty warm incubator. You're going to have water condensation that's going to start building up on the lids, and so you make sure that the agar is faced side up. And usually the bacteria stays in the incubator. We usually leave them from 24 to 48 hours. And that's, most bacteria within 24 to 48 hours will start growing. Okay, and they're going to start growing on the agar, on that growth, and that growth medium that you have within your plates. Right here, those are a bunch of videos that you should go ahead and watch. And there's also a virtual lab that I want you to go ahead and try out for this week. We're going to also go ahead and introduce the different types of media that we use. So as I was just saying, those Petri plates, we go ahead and we fill them up with a growth media. Growth media, it's basically going to contain food, nutrients for your bacteria to grow on. And as I said, to solidify the media, what do we add? To solidify the media, we usually add agar. Okay, and we said that bacteria do not consume the agar. Now, by the way, some of you are going to ask, do we always have to place, do we always have to add agar to our media? No, you don't have to, okay? So if you add agar, your media is going to solidify. If you don't add agar, your media is actually going to stay liquid, okay? And I'm going to show you, later, a few examples of how a liquid media would look like, etc. So we're going to discuss that later. Now I want to talk about the different types of media, okay, that we use. So some of the different types of media that we use, we're going to use Sabdex, nutrient agar, tryptic soy agar. Let's start with the first one. Okay. Now, Sabdex, we classify it as a selective medium. What's a selective medium? A selective medium basically encourages the growth of certain microbes and prevents other microbes from growing. Okay, so it's going to encourage a specific group, okay, to go ahead and grow on that medium, and at the same time it's going to discourage and like prevent other microbes from actually growing on that medium. Okay, Sabdex, when we come and make the Sabdex medium, okay, some of the characteristics that it's going to end up having is that the pH of the media itself is pretty low. Okay, like it's a pH of around like 5 to 6. Okay, and sometimes they can go ahead and even make it lower. It also has pretty high sugar levels. Okay, so which microbes actually love such conditions, and therefore the Sabdex is actually going to encourage their growth? Fungi. Okay, fungi, they prefer high sugar and low pH environments versus bacteria. Bacteria, a lot of the bacteria out there do not grow very well in a low pH environment. So the Sabdex is actually going to encourage the growth of fungi, and it's actually going to prevent the growth of bacteria. So let's say I want to go ahead and culture bacteria. Should I be using a Sabdex plate? No, I shouldn't be using a Sabdex plate. Okay, so when should I use a Sabdex plate? I should use a Sabdex plate if I want to grow fungi. Okay, so if I want to go ahead and culture fungi, which is the best medium for me to use, if I want to culture fungi, the best medium would be the Sabdex. Okay, there's another type of medium called nutrient agar. Nutrient agar is a standard medium. So nutrient agar, it's commonly used. Okay, it's made out of different types of nutrients in there. And it can grow a bunch of different types of microbes. Okay, so again, it's not going to be selective or anything, like a bunch of different microbes are going to grow on nutrient agar, and that's why nutrient agar, it's highly common, right, for us to go ahead and use it. Because let's say you're sampling a surface, and you have no idea, what type of bacteria or, microbes you have on that surface. So it's best to use nutrient agar, since nutrient agar can allow a bunch of different types of microbes to go ahead and actually grow on it. There's another one called the tryptic soy agar, or TSA. TSA, it's what we refer to as enriched media. So it's enriched because it has some extra nutrients. Okay, so it's enriched as a bunch of extra nutrients, and we usually use a media that has extra nutrients when we're trying to grow bacteria that are pretty picky eaters. Okay, what do we call them? Bacteria that are pretty picky in terms of the nutrients that they want. We refer to them as fastidious bacteria. Okay, so if you want to grow fastidious bacteria, those picky eaters, it's best to use tryptic soy agar. Tryptic soy agar is usually used to grow microbes from human skin. But that doesn't mean, by the way, that you can't grow microbes on your skin on a nutrient agar plate. You can, okay? Since nutrient agar, again, allows a bunch of different microbes to grow on it, but tryptic soy agar, it's going to go ahead and especially for those fastidious bacteria, those picky eaters, it's going to be a pretty good medium to use. These are some images that I wanted to go ahead and share with you. So this one right here, it's an example of a Sabdex. Remember how we said Sabdex is selective? What is it selective for? It's selective for fungi. Why? Because it has a pretty low pH, right, and high sugar levels. And so, as I said, fungi, they prefer low pH environments. And so Sabdex is going to be selective for fungi. How do you know, by the way, that this is fungi growing on your Sabdex? Fungi have a pretty unique feature about them. They're fuzzy, okay, like fuzzy, hairy. A lot of them are going to be like white or green or black. Those are like 3 common, colors, but also some of them could be like tan like this. But again, they're going to look fuzzy and hairy, okay? This plate, by the way, what they did was take off the lid, okay? And they left the plate exposed to air for 15 minutes. So all of that nastiness that you guys are seeing right here on the plate was just because you left it exposed for 15 minutes. And this shows you, like, how much there are like fungi, spores, etc. in the air, and they dropped on the plate and they went ahead and they grew. And again, why are they growing? Because this plate, this media, it's providing them with the nutrients that they need, okay? This plate right here, it's the tryptic soy agar. And as I said, the tryptic soy agar, it's an enriched media. So fastidious, those picky eaters, they can go ahead and grow on tryptic soy agar. Now, notice how they don't look like this type of growth. So this is a common look for bacteria. So no hairiness or fuzziness. They kind of look like this. And by the way, these circles that you're seeing, we refer to them as colonies. What's a colony of bacteria? So bacteria, they replicate super fast and then they kind of keep growing and next to each other, and they kind of start like, even on top of each other, and they start creating these, circles that you're seeing on the plate, and we refer to them as colonies, okay? So one colony, by the way, is not one bacteria. One colony, like this colony right here, is going to have tons of bacteria cells within it, okay? So each colony has a bunch of bacteria, okay? And you can see, actually, based on the different colors and sizes of the colonies, that's telling you that there are different types of bacteria, okay? And again, they took samples from the human body, okay? This plate right here, it's a nutrient agar plate. The reason why it's divided in half, because right here they sampled a doorknob, and then right here they sampled the same doorknob, but after disinfecting the doorknob using bleach. So right here, this is the side that was before the disinfection. You guys can see how much, again, bacteria growth you have on it. And then on this side, this is after it was bleached. So, what can you tell based on this observation that you're looking at right now? Bleach is pretty good in killing bacteria. Okay, that's why you're going to see, and you probably noticed that at hospitals, for example, they go ahead and bleach gurneys, for example, in between patients. So you're going to notice that, you know, they're bleaching the doorknobs, like with their, like, you know, they use those bleach wipes to wipe the doorknobs, to wipe the gurneys, to wipe, you know, handles and everything in between patients, because they are, you know, they're trying to prevent contamination. Okay. And as we said, those fomites, those surfaces, you can have bacteria growing on them, such as the doorknob. And so it's a pretty, you know, good practice, especially, for example, in, you know, hospitals, doctor's office, etc. You would find them cleaning after, you know, after each patient, so that when a new patient comes in, they're going to be, you know, lying down, right, on a gurney that's now, you know, that's now been cleaned. Okay. So these are some images that you guys are going to use when you're completing worksheet number 2. Take a look again at, you know, the variety of microbes that are growing on those plates, compare the plates, and that would help you to go ahead and answer week 2 worksheet. And as I said, those are called colonies. Okay, so these are called colonies right here that you are seeing on those plates. And that would be all.